So today I'd like to go over probably the last video in my series about studying for the midterms for biology. So this is just on our most recent unit. I don't know if we're going to have another unit after this, but before the midterms, but if we do, then I'll just make another one. So yeah, I'm going to do this and start. So what does DNA stand for? Deoxyribonucleic acid. What are the building blocks of DNA? Nucleotides. What are the three main parts of DNA? Deoxyribase sugar, phosphate groups, and nitrogen base. Picture DNA in a diagram. So it looks a little bit like this. The circle part, the circle is your phosphate. Oops, accidentally. The sugar is that little thing, and then the square is your base. What are the four possible bases of DNA? Adenine, thymine, guanine, and chitosine. Who is credited for developing the model of DNA? Watson and Crick. Who actually made it, and just as a hint, she got radiation poisoning and died, and they won a Nobel Peace Prize for her work. So what's her name? We should know her name. Rosalind Franklin. Okay. What is the shape of DNA called? Double helix. So what's on the sides of the ladder? So since DNA is like a ladder, like a twisted ladder, what's on the sides of that? Alternating sugar and phosphates. What is on the rungs of the ladder? Nitrogen bases and pairs. What holds the two sides of the ladder together? Hydrogen bonds. What is Chargaff's rule? Adenine goes with thymine. Guanine goes with chitosine. What is the function of DNA? So it contains genetic information in the nucleotide sequence, and the DNA code determines the order of amino acids in a protein. What is a chromosome? DNA wrapped around a histone, which is a histone is a protein. Segments of DNA that code for proteins are called genes. DNA replication equals making a copy of DNA. DNA is replicated at the end of blank, before blank, and blank. So it's replicated at the end of interphase, but before mitosis and meiosis. What are the steps of DNA replication? Number 1, enzymes unzip the DNA by breaking the H bonds. Number 2, enzyme moves along the single strand of DNA and matches up DNA nucleotides with the original nucleotides following the base pairing rule. What does RNA stand for? Ribonucleic acids. What are the building blocks of RNA? RNA nucleotides. Why is RNA not called DNA? Because it has ribose sugar instead of deoxyribose sugar. How else is RNA different from DNA? What's unique to RNA? So RNA has uracil instead of thymine. It's single-stranded, and it can leave the nucleus. What is mRNA? Messenger RNA. How is mRNA formed? Transcription. What does mRNA do? Its purpose. So it carries the genetic code from the DNA into the nucleus. It carries the genetic code from the DNA in the nucleus out to the ribosome in the cytoplasm. So DNA from the nucleus goes to the ribosome. And it copies and carries a message. That's what you need to know. Copies and carries a message. So what is tRNA? Transfer RNA. tRNA is involved in translation. In translation, each tRNA molecule in translation, blank, carries a specific amino acid to a specific place in a protein. What is the purpose of tRNA? Carries the amino acids. What is ribosomal RNA? A major component of the ribosome. DNA contains the code to form all the blank in the body. Proteins. This allows the DNA to make blank. Structural proteins and enzymes. What is transcription? The copying of the DNA code into a molecule of mRNA. What happens in transcription? Number 1, a specific gene on the DNA molecule unzips with the aid of special enzymes. 2, individual RNA nucleotides match up to the DNA nucleotide following the base pairing rule. 3, newly formed mRNA molecules attached to the ribosome. So what are mRNA codons? Groups of 3 nucleotides in mRNA. For example, in transcription, so let's make an example of what it is when we're going to change DNA to mRNA. So G goes to C, A goes to U, G, we said G. C goes to G, and T goes to A. So it switches as usual, except you get a U instead of a T. So what is translation? The genetic code on an mRNA strand is changed into a protein with the help of tRNA molecules. What is an anticodon? tRNA. What are the steps of translation? Number 1, the mRNA code is translated by reading codons. 2, tRNA molecules carry specific amino acids to the ribosome. 3, anticodon with an amino acid match up to the mRNA codon. 4, as the ribosomes move along the mRNA, more tRNA molecules bring in more amino acid to build a protein. 5, a stop codon is reached, translation ends, and a new protein is formed. What is a mutation? It's a change in the genetic sequence of a gene or an entire chromosome. It's passed on to all cells that develop from that gene. Describe point mutation. It's caused by a base substitution. This may or may not affect one amino acid in the protein. Describe frame shift mutation. It's caused by inserting or deleting a nucleotide. This results in many amino acids being changed. What are some examples of gene mutations in humans? Sickle cell anemia, phenylketonuria, cystic fibrosis, and Tay-Sachs disease. How many chromosomes do most humans have? 46. What do sex chromosomes do? They determine the sex of the person. So XX female, XY male. What are autosomes? The other 44 chromosomes that aren't sex cells. What is a karyotype? A picture of a person's chromosomes arranged in homologous pairs according to size. Why might doctors use karyotypes? To see any chromosomal mutations. What are chromosomal mutations? Too many or too few chromosomes. What might chromosomal mutation be caused by? Non-disjunction during meiosis, which is improper separation of chromosomes. Most non-disjunctions are blank. Fatal. I said this very, they're mostly fatal. So, what are some examples of non-fatal chromosomal mutations in humans? Down syndrome, Klinefelter, Turner's syndrome. What's the impact of mutations in body cells? Only damages that particular individual, and it cannot be passed down to their offspring. Like if I have a mutation in my arm, my kid isn't going to have that same mutation unless it's something that I inherited from my sex cells. If that makes sense. Yeah, so like no one else is going to, like if I have like a giant third arm. My kids don't necessarily have to have that third arm. Okay, so what's the impact of mutations in gametes, the sex cells? They can be passed on to the offspring, and it obviously affects them themselves. What are mutagenic agents? Things that can cause a mutation. What are some examples of mutagenic agents? So we got radiation, such as X-rays and ultraviolet rays, and some chemicals like formaldehyde and benzene. All cells of an organism have the same link, DNA. Depending on cell type, different genes are switched on and off. Which genes get switched depends on blank. Cell history and environment and selective breeding. What's an example of an animal getting a characteristic due to its environment? They mention this in the regions for some reason. So, Himalayan rabbit changes its fur color based off of the temperature. Like if you put a block of ice on it, it's going to get a different color skin on that just specific place, fur, I mean. So, what are some examples of gene technology / biotechnology? Recombinant DNA, transgenic organisms, cloning, human genome project, DNA profiling / fingerprinting / gel electrophoresis. What is a genome? All genes on a particular species. What is genetic engineering? It's adding desired traits to an organism's genome. What is plasmid? A small circular piece of DNA found in bacteria, but not found in the chromosomes. So it's just found in the DNA in the bacteria. What are steps used in recombinant DNA techniques? So this is a long one. Number 1, special enzymes called restriction enzymes are used to cut out a piece of DNA. The enzyme cuts the DNA at specific locations, leaving sticky ends, and the same enzyme is used to cut open a plasmid, which will also have sticky ends, 2 sticky ends. And the cut pieces of DNA are added to the plasmid. This is recombinant DNA. 3, the plasmid with the added gene is put back into the bacteria cells. 4, the recombined cells are allowed to reproduce to get many cells, which is cloning. What are transgenic organisms? Organisms with genes from another species. What is cloning? Creating a genetically identical organism. What can we do cloning on? So it's easy to do with simple cells like bacteria and plants that practice mitosis to reproduce, and tissue culturing. Recently, we can do mammals, including sheep, mice, cows, and pigs. So, done a lot of stuff. But what are the steps to cloning? Let me check out what I'm saying. Yeah, what are the steps to cloning? Okay. So number 1, an egg cell from a female has its nucleus removed. So female's egg cell removed. Donor's DNA is taken from the animal that is to be cloned. 3, the nucleus from the clone is put into the egg cell. Typically an electric shock gets the cell to start dividing. 4, the new cell is allowed to divide mitotically to form an embryo. So get an embryo now. 5, the embryo is implanted into the foster mother's uterus where it continues to grow and develop. Now, okay, so what is the purpose of the Human Genome Project? Number 1, to determine the location of all 20,000 genes in a human. 2, to sequence for all 23 base pairs of humans chromosomes. Find the base pairs of chromosomes. So, what is DNA profiling also called? Gel electrophoresis and fingerprinting. What are some uses of fingerprinting? To determine heredity, for paternity lawsuits like who's the dad, who has to pay child support, and for criminology. What are the steps of DNA fingerprinting? Number 1. So you cut the DNA, leaving behind blunt ends using restrictive enzymes. Number 2. You put the DNA fragments into a hole in the gel. 3. Turn on the power and separate the DNA fragments according to size using gel electrophoresis. 5. Use radioactive pulse to label the DNA on the gel. Okay. Last question. Smaller fragments travel blank when you're doing this gel electrophoresis thing. It travels faster. Okay, so I hope that helped. Guess we got through this all. So I'm going to do a similar series with the notes for history. But I hope this helps, and adios.